inviting Spark to talk about a little bit what we do, and my objective today is to put the work and the history of Spark in the context of the history of gene therapy. Just to give you a brief background, I'm the Chief Scientific Officer of Spark. I've been there for a short time, it's probably about 4 months now. I do come from a long history of academia in the field of gene therapy, and in fact I was involved in the early work, which then led to the spin-out of Spark Therapeutics. So this is the forward-looking statements, and by the way, it's great to be here today in this field with the Eagles. I think it's quite exciting. Before I go into the gene therapy, I would like to also put this into the context of the development of a new class of therapeutics, because today we hear a lot about gene therapy, but it is still a fairly new class of therapeutics. And so if you look at the past, of course, gene therapy is somewhat similar to biologics, and we know that the first biologic to be licensed for human use is insulin in the 80s. And really, after that, after this first drug that was licensed, there was a cascade of many other biologics, and now we know today that this class of therapeutics is really important and constitutes a fundamental part of the pipeline of several pharmaceutical companies. And this is what is happening today with gene therapy. And I thought I would put in this slide the drugs that have been licensed so far as far as gene therapy products. There's one quite interesting point, which is that many of the drugs you see on the screen are actually coming or related to the area where we live now, right? Because we have Strimvelis, which was a collaboration between GSK and an academic center in Italy, which was licensed in 2016. We have this past year we've seen 2 CAR T cell being licensed, one from Novartis and one from Kite Pharma. And again, CAR T cells, a lot of the work has been done at the University of Pennsylvania. And finally, I will tell you a little bit about Luxturna, which is the gene therapy drug for a form of congenital blindness. I'm sorry, I'm a little congested, which was licensed this year, was approved by the FDA, and we hope it will be in the next year approved by the EMA as well. So, gene therapy, just for those of you who are not familiar with the concept of gene therapy, consists in using, it's a drug that is made of a DNA or RNA, anyway, a nucleic acid, which is placed into a vector, something to transfer it within a cell. And so most of the gene therapies that we see today, also as drugs, licensed drugs, are derived from viruses. And in the case of the virus that we use the Spark to make Luxturna, this virus is called AAV, and it's a virus that is produced, as you see here, in large quantity, then injected directly into the patient. And really, the idea of gene therapy is that we want to have a single treatment, so one intervention, of course, hopefully achieve therapeutic effect, so correct a disease, and we want that this to last for a long, long time. Okay? Ideally, only one treatment in the life of a patient, and this is the concept of gene therapy and the goal. So to go a little bit in the detail to give you an overview actually of the history of gene therapy, I think this is actually very important because it will help you understand what was the role of academia, what has been the role of academia in gene therapy, and what is the role of industry. One thing that we didn't know at the time was that the technology had some side effects. And so, in fact, it was around the beginning of the 2000s that some very high-profile adverse events were were seen in clinical trial of gene therapy, including the death of a patient in an adenoviral gene therapy trial, which was done at the University of Pennsylvania, and then there were some cases of leukemia in a trial that was done conducted in France. And so this represented an important setback for the field. In fact, the message was that it was just not ready for prime time because there was not enough understood about the technology. And this, at the time, led to a decline of the number of trials, decline of interest from the patients, of course, but also, and importantly, the investment from industry and investors in general really declined. And in this context, really, this represented a major change in landscape. I would say probably all the companies that existed at the time that do not exist anymore, that did not survive to the time of lack of interest from investors in gene therapy, and also all the problems that gene therapy seemed to have. However, yes, the medical need around genetic diseases remain, right? I think that's obvious. Today we know that much better. And at the time, really, everything fell back into academic medical centers, such as Children's Hospital, where the work continued in a number of therapeutic areas. And here I have some of them: sickle cell, cystic fibrosis, but really a lot of the work of gene therapy at that point fell back into the academic centers. And this is very important because in this context, in fact, Children's Hospital of Philadelphia, just to remain local, decided to invest in gene therapy. And in fact, at the time I was there, they decided to create a center which is still there, Center for Cellular and Molecular Therapeutics, which was really like a small biotech focused on cell and gene therapies with all the functions that you may think you need to bring something to early phase clinical trials. And in particular, as you all are familiar, you need to be able to produce gene therapy products in a quality that is amenable for clinical trials, early clinical trials. So there was a GMP vector facility, and also the center provided clinical and regulatory support to the investigator that had ideas that were ready for prime time. So they had a proof of concept that was ready to be translated to human trial. And so this work was quite, let's say, successful because in the years of operation of this center, there were a number of clinical trials where basically it was demonstrated that gene therapy was safe, first of all, and also was able to potentially lead to a therapeutic effect. Now, I don't want to say that all the work was done at the Center at Children's Hospital. In fact, in parallel, there was a lot of work in many clinical centers around the world, which really showed that gene therapy really had the potential to change the life of patients. And I think what is also important is that at the time, all this work was financed, as I say, by the Academic Medical Center, the NIH, Scientific Society. So anyway, there was no industry and no investors, but there were money that allowed really to do the work without the pressure of delivering product, and also without the pressure of maybe going after markets that had some interest, right? And so in fact, and I'll come back to that, some of the products that I showed you in the earlier slide that now are licensed drugs, as you know, they are for fairly rare diseases that perhaps today will be less attractive. The other point that is very important is that during these years, these sort of protected centers allowed the investigators also to work a lot around the issues that were encountered in the technology. So there were a lot of advances, new technology, new vectors that were developed. And in the case of AAV vectors, which are some vectors that some of the vectors that are used for gene therapy, the issue of immunogenicity, for example, was addressed in a quite systematic manner. And so the overall effect, you see it in the last bullet point, really this de-risked the technology a lot, and that really helped the field to move forward. The first two bullet points are a little technical, but essentially we were able to develop safer gene transfer vectors and also address some of the hurdles that were encountered in the early trials. And the results finally arrived. These are some examples of clinical trials where there was demonstrated long-term correction of hemophilia. There were the early trials for gene therapy for congenital blindness. And finally, in the little picture there was the first drug that was approved in Europe for a very rare disease, which is called lipoprotein lipase deficiency. So of course this also it was another big milestone in the field of gene therapy, which changed completely the landscape again. And in fact, the capital moved back, and I think we are getting closer to today, moved back to the field of gene therapy. And, and so this is when the whole discussion about Spark initiated. In fact, at the time, what you see up there, vorticine and parvovirus, so the gene therapy for congenital blindness that was developed at the Children's Hospital of Philadelphia became, it was basically about to enter phase 3. And so the idea was, the main question was how to bring that to the market, because probably bringing to the market a drug is not the job of a children's hospital. And there were a lot of options, and of course, as you know, I'm here because the option that was chosen was that to spin out a company, which is today Spark Therapeutics. So Spark Therapeutics was founded in 2013 as a fully integrated gene therapy company because we have all the functions, we also have manufacturing. It was spun out, as I told you, from the Children's Hospital of Philadelphia, from the Center for Cellular and Molecular Therapeutics, and had technology that were developed at CHOP, at UPenn, and also in other institutions in the United States. There was a manufacturing facility which still exists there, and we have one at Spark. And really, this was to support not only the phase 3 for the gene therapy for Luxturna, for gene therapy for congenital blindness, but also other programs that were ongoing at Spark, including one for hemophilia B, which is done in collaboration with Pfizer. Today, we're working in several areas, so we grew a little bit also in our ability to develop gene therapy products, and there are three main target areas. The first one is the eye, and this is where we have our first approved product, Luxturna, along with other gene therapies. Then we have a series of products in development for let's call liver-directed diseases, but it's really where we're targeting the liver to correct the diseases, and finally we are active in the field of neurodegeneration. Just a couple of more minutes, I want to get a little bit more give you a snapshot of the work we've done on gene therapy for congenital blindness caused by allelic mutations in a gene that is called RPE65. As I told you, this is a good example of work that started in academia. In fact, the very first studies where there was evidence that it was possible to treat a disease of the eye, a genetic disease in the eye, were done in the early 1990s. And the early clinical trials, phase 1-2, were also conducted in academia at the Children's Hospital of Philadelphia and University of Pennsylvania. And finally, as I told you, the late work was done at Spark. And this is just an example of the results. I think they were quite impressive, were essentially, the following gene therapy, and basically you can look at the green line, which basically represents the ability of the treated patients to move in an environment in conditions of low light. So there was a gain in function, in visual function, in all the patients treated, and the blue line here will represent the control group, which then, once they were treated, they really gain again visual function. Just to tell you that all the results were quite compelling, and in fact, this is why one of the reasons why the FDA decided to approve this drug in December of last year. So this is just some more latitude on the phase 3. I think the treatment turned out to be quite safe. There were some adverse events, but mostly because the therapy involved a surgery in the eye, so there were some surgery-related side effects. And this is some labeling relative to the drug, but I think I can skip that. And finally, the last few minutes is after the success with the eye gene transfer. I think the next wave of work, not only as Spark, but in the whole field, is to target larger organ, larger indications, and one of them is, of course, hemophilia. Again, a lot of the work that we know today is ongoing in companies, was initiated in academic centers, but actually this is a good example. The very early proof of concept came from a company, Avigen, which then disappeared, and then when all the work went to academia before then going back to the industry. So I think in interest time, this is some of the results which may be interesting to show the power of gene therapy. I think it's always, even though I'm a scientist, I'm always quite amazed by looking at the data where essentially these are patients who received a single treatment, and the green line basically represents their factor levels, their activity levels of factor 9, so correction of the disease at this point 20 months after gene therapy with no further treatment. And so this, I think, demonstrates the potential of this therapy. Of course, this is an example. Today in the field, we're seeing more and more of such examples of long-term correction of several diseases with gene therapy. These are all phase 1-2 trials, of course, and so the key will be to work to bring them all the way to phase 3 and then licensing. This is just another piece of data showing how in these two graphs, in the graph on the left side, you can see how the green lines represents the patients before treatment, so they were bleeding because they had hemophilia, and after treatment, essentially, there was complete correction of the disease, no bleeding, with no further treatment other than the infusion of the vector. So I think, okay, this is work that was done at Spark in collaboration with Pfizer, but I think again gives a good idea of the potential of gene therapy in terms of changing the life of patients. This is just a slide about adverse events, which was quite favorable. There were minimal adverse events, and in fact, other than one episode of asymptomatic transaminases elevation, was nothing else related to the product. And as I say, the results are quite promising. Today we know we can use gene therapy to correct hemophilia B, but also there's good data coming for hemophilia A, so another genetic disease. And I think there's a lot of work. I'm going to say that the key will be in probably the last bullet point of my slides, which continue to expand this observation into larger cohort of patients to confirm the initial findings, of course, in the early phase trial, and then also to bring these therapies to all the way to licensing. And then just my last slide, I promise. Of course, one small comment about manufacturing. We've done a lot of, we put a lot of effort in manufacturing. Today, in the company, we have 2 manufacturing suites, which also serve the commercial phase for our product Lusterna. And then, of course, like many other biotech companies working in the field of gene therapy, we're thinking about scaling up production. And for that, at the moment, we're collaborating with a CMO for our scale up. And then I'm going to close here by saying that, of course, I think in a few words, I told you how gene therapy really started in a place where there was a lot of excitement, and then this excitement faded out because of problems that were encountered. And I think that was the moment where academia helped the field to move forward, and now we're back to a reality of investment, excitement. I think, and I hope, this is going to continue. And this is really necessary because we want to bring this therapy to patients in the form of drugs, so we need the industry. One thing that is important is that, of course, every day we read about new technologies, and it's all very exciting, and I think a lot of them have a great therapeutic potential, but of course, I still think that the interaction between the two parts, the academia and the industry, will be important, because the academia sometimes has been more effective in addressing perhaps some of the hurdles of these new technologies by going a little more details into the mechanism, and so I think the interaction should continue. And then I'm closing here, and I thank you for your attention.